students, now we'll move on our discussion towards the first high-level grouping of the kingdom Animalia, that is the phylum Porifera. These are the simplest organisms present on the entire earth, that is, they have cellular level of organization. So we'll start off with the major characteristics that these animals share. So let's start. So now we're going to start with the various high-level groupings of the animal kingdom, and among the 10 phylums we will be starting with the first phylum, that is the phylum Porifera. Phylum Porifera includes the sponges which have cellular level of organization, are radially symmetrical animals, and are diploblastic animals also. So in the phylum Porifera, the entire body of the phylum Porifera is covered with numerous mouthlets that are also known as ostia. These are the pores present on the body of the sponges. And there is a single opening at the top of the sponge body which helps in the exit of water and particles from the body of the sponges, that is known as the osculum. The body wall of the sponges, it consists of two layers, the outer pinacoderm and the inner choanoderm. Between the pinacoderm and the choanoderm, there is a distinct gelatinous matrix called the mesenchyme or the mesohyle present between the pinacoderm and the choanoderm. It acts as an interconnecting layer between the pinacoderm and the choanoderm. So this layer is called the mesenchyme or the mesohyle. Now the mesenchyme consists of skeletal elements or free amoeboid cells. Here in this, you can see the basic sponge body plan, in which there are numerous ostia or the pores you can see on the body of the sponge. There is a characteristic, the body cavity of the sponge is known as the spongocele, and through spongocele, the cavity for the exit is known as the osculum. So water entering through the ostia enters into the spongocele, and through spongocele it is taken out through the osculum. Here in this diagram you can see the mesenchyme layer here present between the outer body wall, the pinacoderm, and the inner body wall, that is the choanoderm. The sponges have several cell types. These cell types are like, the first is the choanocyte, which is also known as the collar cell. These have flagella on them and they function as the sponge's digestive system. So the collar cells are composed of microvilli which are present on the collar part of these cells. These are used to filter the particles out of the water. The next cell type is the porocytes, which are tubular cells, and they make up the pores on the body wall of the sponges through the mesohyle. They help in the water passage. The next cell is the pinacocyte, which forms the pinacoderm. The outer epidermal pinacoderm is the outer epidermal layer of the cells. The modified pinacocytes are known as the myocytes. The next cell is the archaeocytes, which are also known as the amoebocytes, are the totipotent cells which can transform into sclerocytes, spongocytes, and collencytes. Totipotency is the property of a cell in which the cell can divide and produce all kinds of differentiated cell in an organism. These archaeocytes also play a very important role in nutrition, nutrition and sexual reproduction. In this diagram you can see the arrangement of the various cells that are present in the sponge body. In the hair, a spongocele and osculum is shown. In the osculum is surrounded by myocytes, tufts, or it may be covered by grate also. and it is excellent in nature. The pores, which are made up of the porocytes, they are inhalent in nature, which take up the water, and it through the porocytes or the pores it enters into the spongocoel, and then it is exited out of the osculum. The pinacocytes form the outer body wall, that is the pinacoderm of the sponges. The choanocytes are the collar cells, which have a tuft of cilia at the collar part of these cells, and a single flagellum is present in each cell. The spicules may be monoexon, triexon, tetraexon, hexaexon, etc. And the last but not least is the amoebocytes, which are divided into several subtypes. The archaeocytes help in sexual reproduction and nutrition. The sclerocytes, they secrete spicules. The spongocytes, they secrete spongin. Collencytes, they secrete the collagen, and lophocytes are highly mobile cells. The body wall of a sponge encloses a large cavity, which is known as the spongocoel, and it is also known as the paragastric cavity. The choanocytes with the flagella is lined in this spongocoel. The most important characteristic feature of sponges, on the basis of which these sponges are divided into different groups, is the canal system. The canal system helps in nutrition, respiration and excretion, and canals are developed due to the infolding of the inner wall. So there are basically 4 types of canal system. Asconoid, which is the simplest canal, it is found in Leucosolania. Syconoid, which is found in the Scypha. The next is the leuconoid, which is the most complex and efficient canal system among the sponges. It is found in the Euspongia. And the next is the rhagon, which is only found in the larva of Demospongia. So this diagram shows the arrangement of the body wall of a sponge, and also showing the canal system. In this, there is shown a radial canal which is lined by the choanocytes. The incurrent canal is lined by the pinacocytes. This shows the incurrent canal. There are numerous prosopiles and apopiles. So these are the 2 important terms with respect to the PMT examination. The water leaves the incurrent canal and enter into the radial canal through the prosopiles. And water leaves the radial canal and enter into the spongocoel through the apopyle. Our next diagram shows the arrangement of the body wall of the sponges according to the canal system of these sponges. The first diagram shows the asconoid canal system, the next shows the syconoid canal system, and the next is the leuconoid canal system. The asconoid, as we have already discussed, that it is the simplest canal system among the sponges. So the pathway of water in asconoid canal system is from the numerous ostia. The water enters into the spongocoel and then into the osculum. It is exited out. The pathway of water in the syconoid canal system, the water enters through the ostia into the incurrent canal. From the incurrent canal into the spongocoel, and from the spongocoel it is transferred to the osculum and exited out. The leuconoid canal system, which is the most complex and the most efficient one also. Here the water enters through the ostia into the incurrent canal, from the incurrent canal into the choanocyte chamber, from the choanocyte chamber into the excurrent canal, and from excurrent canal into the osculum. The spongocele is not very distinct in the leuconoid sponges. The skeleton in the sponges, the internal skeleton, it consists of tiny calcareous calcoblast or siliceous spicules. The digestion in the sponges is intracellular and it occurs in the food vacuoles of choanocytes which help in the digestion in the sponges. The cells which store the food in the sponges are known as the thisocytes. The sponges lack nervous system and they have a great regenerating power due to the archaeocytes which are totipotent. So this is an enlarged view of collar cells. Here, a flagellum beating movement of the flagellum helps to force out the water of the large osculum and pull water in through the ostia. From here, the food particles, it enters into the cell body of the food vacuole, cell body of the collar cell which is stored into the food vacuole, as you can see here. So particles of the food are drawn into the collar to become trapped and then digested by the food vacuoles, and they are engulfed into the cell body and then digested into the food vacuole. So student, we just finished our discussion about the general characters that are shared by all the porifers and how the sponges have been classified on the basis of the water canal system and how the processes of digestion and respiration takes place in the sponges. So in the first part, the general characters were covered. In the next part, we'll be covering about the reproduction, how the sponges or the porifers reproduce. So stay tuned for that.